In this video, I'm going to talk about genetic drift in populations. So genetic drift involves random evolutionary change, and it's a term that describes changes in the allele frequencies in a population due to random events. So just by chance, in each generation, some individuals will leave behind more or fewer offspring, by which I mean they'll leave behind more or fewer alleles or genes. So for example, in this group of ladybugs, some are red and some are green, a random event could prevent certain individuals from surviving to reproduce. And this is going to impact the allele frequencies and the genetic diversity of the population and changes the likelihood of certain alleles being passed on to the next generation. And this happens through a random process. So there's no natural selection occurring in this particular situation. So if the population is quite small, then the impact of genetic drift is greater. So remember that genetic drift is a random process that causes divergence among populations over time, and this is most easily visualized with island populations. So here we have 5 islands, each with 10 diploid individuals, which means that each individual has 2 copies or alleles at a particular locus, which in this case is at a neutral locus, meaning that it's not under selection because it doesn't impact any particular traits. In this example, we have allele A1 in blue and A2 in red. So at some initial start time, we call T0, all of the individuals are heterozygous on all 5 islands. So if we counted up all the individuals, we'd have 50 individuals, and all of them have a copy of each of the 2 alleles. So the observed heterozygosity is 1. At some point later, T1, there's been a shift in allele frequencies that's causing a shift in the observed heterozygosity. So in this particular case, only 19 of the 50 individuals on all 5 islands are heterozygotes, and that gives us an observed heterozygosity of 0.38. Even further along in time, at time T2, there's been even more of a shift, and some of the alleles have become fixed for an allele. So if you look at this particular island, you can see this island has become fixed for allele A1. This island has become fixed for allele 2, and really the only island that has any heterozygotes or contains both copies of the allele is this island here. We can see down here we have an A1 and an A2 and a heterozygote and a second individual here. So if we want to look at the allele frequencies, on this island at time T0, all the individuals are heterozygotes, and if we counted up all the number of A1 alleles and divided it by the total number of alleles, we'd figure out that the allele frequency of allele A1 is 0.5. And similarly for A2, it's 0.5 as well because it has to add up to 1. In this particular case, all of the islands have allele frequencies of 0.5 for both alleles. At time T1, we see there's a shift in allele frequencies. Now this island has stayed the same. There hasn't been a shift at time T1. But if we were going to look at any of the other islands, we see there's been a shift in the allele frequencies. Not quite as much as this first island, but they are starting to change. And then again, at time T2, we see that the allele frequency for A1, as we mentioned before, has become fixed in this population, meaning the frequency of that allele is at 1 and the frequency of the A2 allele is 0. And similarly, for 4 of the 5 populations, they have become fixed for an allele. And the only way to get the other allele back into that population would be through a migration event. For example, if an individual came from this island over to this island and introduced the A2 allele through gene flow, then that allele would be reintroduced into that population. So that's a brief look at genetic drift and how it changes allele frequencies in populations over time.